Good morning or afternoon, everyone. It looks like my webcam may not be showing. Not sure why that is now. I've had some issues occasionally, but so apologies for that. Welcome to day 3 of the Western Regional Panel on Aquatic Nuisance Species virtual 2020 meeting. We're glad you can join us today and hope that you've been enjoying the earlier parts of the week and can hang with us as we continue through the rest of the week. Today's session is on invasive aquatic plants, and we can consider those both freshwater and marine algae plants. And as you're probably aware, invasive aquatic plants are a widespread issue in the West, and they have high impacts, and there's typically a lot of management activities undertaken, though obviously that can be very variable. Aquatic invasive plants have considerable impacts on the environment, as well as our infrastructure and even aesthetics. So they are known to modify flows, increase sediment, create cover for predators, and just really a long list of impacts. And it's likely that we'll continue to have a lot of invasive aquatic plant issues in the West as we continue to have environmental modifications and the potential for climate change and continued and ongoing release and spread of aquatic invasive plants. So as with all aquatic invasive species, there's many challenges to treating invasive aquatic plants in aquatic environments. And today we'll hear from several of our colleagues throughout the West who have experience with some of the tried and true methods, as well as looking at new technologies that may assist us in our future management. A couple of housekeeping information before we move on to the presentations. If you have questions for our presenters, please put those in the questions box in the GoToWebinar and indicate which speaker that question is for. And at the end of all of the presentations, we will bring up all of the speakers on a panel and get through as many questions as we can. And our speakers have committed, if we're not able to get to those questions in the time we have allotted today, that they will follow up on those questions as well. Also, we've found that some of the audience have had their screens freeze up during this 2-hour session, and that has seemed to be solved by refreshing your browser. So it seems to time out. So if you have that issue, please give that a shot first to see if we can't resolve the issue. And I think that's it on the housekeeping, unless Mason, you'd like to add anything in addition?
That about covers it for now.
Okay, great, thank you. All right, so we can get into our first presentations. And our first presenter, there's going to be a little change to the agenda order. We're going to begin today with what was scheduled to be the second presentation by Dr. Bradley Sartain. And Dr. Sartain, and I apologize if I mispronounced your name. Dr. Sartain has been studying invasive aquatic plants for the past 10 years with a primary focus on their chemical control. Dr. Sartain earned his Bachelor of Science in Wildlife Fisheries and Aquaculture in 2011 and Masters of Aquaculture in 2014 from Mississippi State University. He went on to work as an aquatic herbicide applicator and fisheries biologist before continuing his education at Louisiana State University, where he earned his doctorate in agronomy in 2018. Shortly thereafter, he began his career as a research biologist in the Environmental Laboratory at the U.S. Army Engineer Research and Development Center. Dr. Sartain's current research focuses on linkage of herbicide exposure with water exchange processes, evaluation of newly registered aquatic herbicides, and the selective control of invasive aquatic plant species. Thank you, Dr. Sartain, and I will turn it over to you.
All right, I appreciate it. Can you hear me?
Yes.
Everyone? Cool. What about presentation view?
You're currently in the
non-slideshow view?
Yeah, you need to put it in the presentation mode.
Let's see. How's this?
Perfect.
Title slides up there and everything?
Yes.
Excellent. All right. Well, I appreciate the introduction and the opportunity to participate in the WRP conference. I am a research biologist for the U.S. Army Corps of Engineers at the Engineer Research and Development Center in Vicksburg, Mississippi, where I specialize in the management of aquatic vegetation, primarily the chemical control aspect. This presentation today is going to cover some of the flowering rush research we've been conducting over the past couple of years on the Columbia River and highlight some of the challenges with managing invasive aquatic plants under high water exchange conditions. Well, so basic outline of the topics I plan to cover today is shown here. Prior to the discussion on the flowering rush research, I wanted to introduce the Aquatic Plant Management Team and highlight our capabilities and the types of services that we provide. So the Aquatic Plant Management Team is comprised of several researchers at both the U.S. Army Corps of Engineers at Erdick and Vicksburg, Mississippi, and remote locations. Myself, Drs. Kurt Getzinger and Dr. Nathan Harms are located in Vicksburg, Mississippi. Drs. Chris Mudge and Ben Sperry are located at LSU in Baton Rouge and the University of Florida in Gainesville. Our team mission is to provide research and development services to selectively manage nuisance vegetation in public water bodies and wetlands. Although we have several different research and development focus areas, the presentation today will focus primarily on the chemical control aspect. So our responsibilities as far as chemical control include in-house research and development to improve US EPA-registered products for managing invasive aquatic vegetation. This may include refining current management strategies for historically problematic species, such as hydrilla, Eurasian water milfoil, water hyacinth, but also developing new management strategies for new invaders. We also serve as subject matter experts on chemical control issues for resource management agencies and collaborate with registrants to develop and evaluate products for use across the U.S. Typically, we follow a from the laboratory to the label approach that begins with small-scale testing, followed by field verification for developing aquatic plant management protocols and management plans. Just some examples of our in-house facilities we utilize for small-scale testing. These include environmental growth chambers, greenhouses, outdoor mesocosms, and outdoor experimental ponds, where we collect this baseline data. And although small-scale research is really beneficial, really verifying that data in the field is very critical to provide guidance at the operational level. We also conduct field studies nationwide from Lake Okeechobee in southern Florida to the Columbia River in Washington State. And these studies are very important because each water body is so unique. So you may have the same problematic plant in two water bodies that are less than 10 miles away from each other, but because those systems are too different, they may require two completely different management approaches to be effective. So that's just a little bit of background on the Aquatic Plant Management Team. I'm going to go ahead and jump into flowering rush control and hydrodynamic systems. Most of you, I'm sure, are familiar with flowering rush. It is spreading throughout the Pacific Northwest. It has a really dynamic ability to grow as an emergent plant in shallow areas, but it can also grow submerged in waters upwards of 18 foot deep. It can form monotypic stands in both of these growth habits, and really negatively impact native plant communities, fish wildlife habitat, water use. And currently there are limited strategies for effective long-term selective control, particularly in hydrodynamic systems, which hydrodynamic kind of relates to a run-of-the-river reservoir. It might be enclosed or impounded by 2 hydroelectric dams where you get a lot of water flow through that area. So a study site for this research, Columbia River on the McNary Pool, or Lake Wallula. Priest Rapids Dam is on the upstream end with McNary Dam on the downstream. Has a drainage area of about 214,000 square miles, and flowering rush first documented 2008, I believe, right around the confluence of the Yakima River, just north of Kennewick, Washington. So in 2018, when we began this research, we started with several water exchange evaluations. This allowed us to get a good comprehensive of what type of flow regimes we were dealing with. So we wanted to pick several different sites that varied between high, moderate, and low flow sites. Based off that water flow, it really impacts what type of management approach you need to take. 4 of these sites, we didn't have any barriers or curtains in place. We did select one of these sites, Clover Island here, to evaluate bubble tubing as a potential method for reducing water exchange. And we evaluated that at a number of different configurations that I'll touch on in the next couple of slides. So for those of you who are unfamiliar with water exchange evaluations, we use a rhodamine WT dye. It's an inert fluorescent dye that we can put in the water column. Most often at 10 parts per billion as the target concentration. We identify a potential treatment plot, outline that plot, put out a series of predetermined points, and go back and sample that dye at specific time intervals. One good thing about the rhodamine WT dye is it's cheap, non-toxic, inert dye, but it really mimics a typical submerged herbicide application, as you can see from these images here, it being put out in a research plot. By taking readings at specific time intervals, we can really get a good idea of the type of water exchange in that area, which can really guide us on what management approach might be the most effective. So some background on the bubble curtain. Bubble tubing is produced by a Canadian company called Canadian Pond Products. It's basically just a durable, flexible aeration tubing. You can see 3 different sizes here on the bottom right photo. Typically in aquatic plant management operations, you'll see people use barrier curtains in high water exchange areas when they're treating small isolated populations of invasive plants. They're a little cumbersome. A lot of them are yellow, if you're familiar, and they have a curtain that drops all the way down to the lake bottom or river bottom. One of the benefits of the bubble curtain compared to the traditional curtain is there's no impact to navigation or boating. Oftentimes with aquatic invasive plants, you get initial infestation at a boat ramp or a marina or something like that. And putting up a traditional barrier curtain can cause some issues because there can't be any navigation and it impacts boating. Also, the bubble curtain is pretty easy to deploy. It lies on the lake or river bottom. You know, with a traditional barrier curtain, if you're doing a substantially large area and a storm comes up, it can really cause some problems as far as twisting it all up, ripping it at the seams and things like that. And with the bubble tubing, it's durable. You can leave it in place. If you need to do a treatment 1 week and want to do a follow-up treatment, say 16 weeks down the road or a year or 4 years down the road, you can leave it in place, come back, hook it up to an air compressor, and be underway. So we evaluated 2 different configurations of the bubble curtain in 2018. For our first evaluation, we had a very close curtain spacing utilizing a single compressor here on that right side of that left picture. This produced airflow about 0.31 CFM per foot. For our second evaluation, we increased the curtain spacing and we added an additional compressor on the upstream end, which increased airflow to 0.62 CFM per foot. Note the arrow of the river flow here and the wind directions between the 2 evaluations. During the first evaluation, we had very favorable wind. Second, not so much, blowing directly into the river channel, where if this was an operational treatment, we would have delayed the treatment if herbicide was being applied because of the unfavorable conditions. But because we were using dye, we went ahead and went forth to the evaluation to see how the bubble curtain performed in less than ideal conditions. Here's just some visual images of what the bubble curtain looks like in operation. The compressors that were used, shown here in the middle picture, just some action shots of it being assembled, and what the dye looks like once it's applied to the plot. It's pretty concentrated. This is actually just 12 ounces that was used to achieve that 10 ppb concentration within the plot. So a little bit of rhodamine goes a pretty long way when you're only achieving a 10 ppb target concentration. Jumping right on into the results, prior to the bubble curtain evaluation, we did do a water exchange with dye and no curtain. Based off this, we had a dye half-life of 3.8 hours. So in that particular scenario, with a favorable wind, we probably would have had suitable flowering rush control with a dicot application. Dicot's often used when you under very rapid water exchange scenarios. But with the bubble curtain during the first evaluation with a favorable wind, we almost doubled the half-life of the dye. And even so, with the second bubble curtain evaluation and an unfavorable wind, we still almost doubled the dye half-life. So even in a worst-case scenario, like under the second evaluation, that was suitable for a dicot application, but also put us in the ballpark of potentially being able to use Invafol, which is another contact herbicide that requires a little bit longer contact time than dicot does. So not only did bubble curtain decrease the water exchange, which would have led to improved control if we were to do a dicot application, but it also gave us the option for multiple tools to be used in the event that we needed to use Invafol or another product. So jumping on to 2019, we continue to do bubble curtain evaluations, although at a different site. For this site, we picked Port Pasco or Osprey Point. Reasons for choosing the site was we had data from 2018 with no bubble curtain, and it has a large stand of flowering rush, which made it a suitable site for an herbicide evaluation as well. So we applied the rhodamine WT dye and herbicide as a tank mix simultaneously to the treatment plot at 4 in the morning on July 30th, 2019. We had a double bubble tubing, it's kind of a tongue twister, arrangement of the curtain around the plot. You can see here in this photo on the bottom right. We were using Dicot Reward as the trade name of the product with a target concentration of 0.37 parts per million. And McNary Dam discharge at the time of application averaged about 149,000 cubic feet per second. So pretty good amount of flow, certainly not the greatest amount of flow that I've seen come out of McNary Dam, but definitely a challenge to get the contact time we were hoping for with that type of flow coming from the dam. So just look at a comparison from our 2018 data at the same site with no bubble tubing. We got an estimated dye half-life of 1.29 hours, which is essentially about an hour and 15 minutes. A little on the fast side. In 2019, with the bubble curtain in place, we extended that by about 30 minutes. Now, this is a site that's right on the edge of the mainstream river. You can visually see the current pushing past the area. So water's moving pretty fast, but we were able to extend or decrease the water exchange for about 30 minutes in 2019 compared to 2018, which this can vary due to different dam operations and reservoir operations also. So jumping into the results of herbicide efficacy, how did our treatment do? Well, this was our plot prior to treatment. Following treatment, we were able to see flowering rush injury start to develop by as little as 2 days after treatment. We did vegetation assessments at 4, 8, 10, and 52 weeks after treatment. You can see here, this is 4 weeks after treatment. You can see the flowering rush in the plot showing some pretty severe injury. The water level's down about a foot or foot and a half. There's still some emergent plant material above the water line. But fast forwarding to 8 weeks after treatment, we actually saw a 40% reduction in flowering rush. There was no emergent plants present. However, we were seeing some regrowth in the site, which is very common when you're doing a contact herbicide treatment. This year, we got to do our 52-week after treatment survey, so 1 year post, and flowering rush percent occurrence was still 90% in the plot. So really, we got a 10% reduction, which is not surprising because in aquatics, typically you need more than one treatment to get the type of control you need. But one surprising thing was there was no emergent plants present in the plot. So that was a great sign that we put a pretty big dent in the flowering rush at this particular site. But like I mentioned, multiple treatments are usually necessary to achieve the type of control we were looking for, so we also applied a follow-up treatment as well. Interesting with flowering rush, in systems like this where you have water flow moving as fast as it is and you have that limited amount of contact time, you're really limited to contact herbicides which don't translocate actively throughout the plant. So the methodology here for getting long-term control is because particularly the triploid flowering rush grows from rhizome, and it'll get established in these areas and have a pretty good rhizome base. Well, it senesces in the fall, but during the growing season, it produces the emergent shoots to reach the water surface where it stores energy and translocates that down to its rhizomes. So by coming in and doing a contact herbicide treatment and getting a good burn down of those shoots and above ground material, you're kind of disrupting its storage capacity to come back the following growing season. So once you burn it down, it has to use energy from those rhizomes to regrow back to the surface within the same growing season. So you can really use up some of those carbohydrates and starches down there in the rhizome, which can facilitate better control in the future. So in conclusion, based off our 2018 results, bubble curtain was successful at slowing water exchange, which potentially would improve herbicide efficacy and also lead to potential herbicide options. Dicot treatment was successful at reducing flowering rush, by 40% 8 weeks after treatment. Only 10% the following year when we did our 1 year post, but there was no emergent plants in the plot. So that was a good sign. Eurasian water milfoil and curlyleaf pondweed were also present in the plot. You can see from pre-treatment percent occurrence, we controlled those, and there was a pretty good reduction there. Looks like the curlyleaf's trying to make a little bit of a comeback at our 1 year post. Our native plant occurrence decreased initially, which is not uncommon when you're doing a contact herbicide treatment. Selectivity is kind of impacted there because of the tools you have to use. But what we typically see in most situations is when you remove the pressure from that invasive plant, you start to see the native plants rebound pretty quickly. So we're starting to see that. I think 0 weeks after treatment, there's about 25% native plant occurrence. In our 1 year post, we were showing 23% native plant occurrence. Unfortunately, for our follow-up treatment this year, due to the impacts of COVID-19, we were not able to do a bubble curtain evaluation this year. So we just had to do a treatment without a curtain and water exchange study without a curtain. But we will be doing our post-treatment vegetation surveys over the next several weeks, and can't wait to see those data, see what type of results we got after a second follow-up treatment. With that, I'd like to thank everybody that participated in this project. Damian Walter was a huge help. The Pasco office there in Pasco, Washington, there's a lot of interesting things that are involved as far as running those compressors and having access to personnel that can help put out barrier curtains and bubble curtains and boats and things like that. And they were tremendous. A lot of work was involved. Aquatechnics was on call to do a dye application, herbicide application whenever we need it, and Canadian Pond for supplying the bubble curtain, and everyone else involved. Is a lot of work involved, but without y'all, it wouldn't have been done successfully. And I want to thank y'all for that. And I might have a few minutes for a couple questions.
Well, we're actually going to hold all the questions till the end of the session. So hang in there, and for our audience, if you have questions, please enter those into the question box and indicate that they are for Dr. Sartain, and we'll get to those at the end.
Awesome.
Thank you very much.
No problem.
And apologies for shorting all of our presenters the few minutes before we started to queue up your presentations and your video cameras. That was my fault. So thanks for swinging with us. All right, up next, we have Christine Jurek and Kylie Couture from Minnesota Department of Natural Resources. And in addition, we also have a late addition to our agenda of Keegan Lund also from Minnesota Department of Natural Resources, who will be joining us during the panel discussion at the end and the questions. So in terms of introducing Christine or Chris and Kylie, Chris is a senior invasive species specialist for the Minnesota Department of Natural Resources. She is working on starry stonewort in Minnesota since its introduction in 2015. Kylie, also with the Minnesota Department of Natural Resources, is a natural resource specialist. She's been working with the invasive species program for over 6 years and has been part of the Starry Stonewort monitoring and management project in the Metro. And we'll start off with Chris. Welcome, Chris, and thank you for joining us today.
Thank you. Can you hear me? Okay.
Yes.
Okay. And then I just want to make sure my PowerPoint is where it's at. Are my notes being shown here, or do you see the full screen?
Your notes are showing.
Okay. Let me flip screens then. Is that better?
Perfect. Take
Okay.
it away.
All right. This is my first virtual conference, so I apologize if anything goes. Let's see. Still can see my screen?
Yes, perfect.
Okay. Okay, good afternoon, everybody. I am Chris Jurek. I'm with the Minnesota Department of Natural Resources. I'm one of 8 invasive species specialists for the state of Minnesota. Today, and my colleague, Kylie, will be talking about 2 of our projects that we manage within the state of Minnesota. I just want to give you a brief overview about starry stonewort. It is a freshwater green algae and it's found in the plant kingdom. There's some key identifiers that I want to point out. It includes the star-shaped bulbils. It has this smooth stem, much like a lot of the native stoneworts that you will encounter. It has a forked branchlet, so you have these uneven bracts on one of the branchlets that is a way to determine if it's starry stonewort or like your native stonewort. And it also has branchlets in whorls of 6. It's a really robust plant, so it's really different than most your native stoneworts that you may encounter. It is an aggressive species, so in both managed and unmanaged plots in Minnesota, we see this species taking over these native plant communities. It spreads by bulbils, which each node actually will form a bulbil, and it also spreads by fragmentation. Starry stonewort was discovered in Minnesota in 2015, and since then, it's in 7 counties and 16 lakes. Over half the lakes are managed by various lake groups and local units of government. Some of these infestations range from less than an acre to lake-wide distribution. Today, we're going to touch on some of our starry stonewort pilot projects. Since 2015, the Minnesota DNR attempted to control some of these new infestations by taking some novel approaches to removing the biomass and limiting the spread. In the upper left-hand corner, that's a picture of a suction dredge. This unit was used to remove both the sediment and the biomass present at the access. We did put up a barrier for this project. So that's one strategy that we tried. We've used pesticide combinations with hydrothal 191, diquat, and copper formulations. We've tried mechanical harvesting, diver-assisted suction harvesting, hand pulling, and or a combination of all methods that I just described. So our objectives, like many invasive aquatic plant management projects, we wanted to see how effective these methods are on the removal of starry stonewort. We monitored these projects using point intercept survey grids at either the management area or lake-wide, focusing on presence and absence of starry stonewort, also percent occurrence. We looked at biomass measurements before and after treatments, and we also used just plain old snorkel and scuba diving surveys to observe change before and after management. We also evaluated impacts to native aquatic taxa, and this includes native plants, including your native chara, which is also a macroalgae that is also impacted by pesticides that we used. One project we did sample macroinvertebrates pre and post copper treatments. That won't be discussed in this presentation though today. We also did some monitoring of bulbils in the sediment pre and post copper treatments over a 2-year period as well. Again, I'm not going to touch on that study, but we didn't see any correlation between copper treatments and bulbils found in the sediment pre and post treatments. We also looked at concentration exposure time of algicides on starry stonert by monitoring copper concentrations. I'm just going to jump into the case study that I'm involved with in central Minnesota. This is in Grand Lake, which is an hour-and-a-half northwest of Minneapolis-St. Paul area. This is a really small lake. It has really good water clarity, and it's about 650 acres in size. Back in 2017, we had a volunteer program through the University of Minnesota called Starry Trek. These volunteers successfully identified starry stonert at the access. Once the initial discovery was found, we worked with the lake group and our local units of government to do a hand removal project at this access. So why did we decide to pull starry stonert? Well, Grand Lake has a lot of native stonerts at the management site, and we felt that the pesticide treatments would not be a good choice because it's not selective. Copper does damage some of the native charophytes that are in this lake. We also wanted to remove the attached bulbils on the starry stonert. Many of you may know that hand removal of these small populations of other invasive plants have also shown significant reductions in other projects in the US. And it's just an opportunity also to learn and adapt a new technique that may be effective at removing starry stonert. So I'm just going to quickly go through kind of the process of how we did the hand pulling at Grand Lake. We first identified the locations of starry stonert, and so in the picture on the left, we threw out some buoys. We had divers. We had 3 divers. We used mesh bags or seed bags for removal. So as the divers went down to the bottom to remove the starry stonert, we placed the starry stonert in the seed bags or lingerie bags to contain the fragments. This lake has good water clarity, so it made it a good candidate for diving. You can also snorkel if water depths are shallow enough to scuba dive or snorkel. And the sediment should be soft, so it's easy to remove the starry stonert. So how do we hand pull starry stonert? There's kind of 2 different methods. It's kind of what's trial and error for us. Basically, you go down and you can grab the plant, or it's not really a plant, it's a macroalgae, but you kind of want to take the sediment, and so you can get the bulbils and the rhizoids. It's slightly attached to the sediments, the starry stonert is. You can pull those up, place them in a mesh bag, and then you send it to the top to the people that are retrieving the plants in a canoe. The other method would be kind of rolling up these beds of starry stonert, and I kind of explain it as taking a sleeping bag, rolling it up, and stuffing it into your bag, your seed bag. It actually works really well. It kind of acts like a carpet on the bottom of the lake, so it's almost easier to remove these dense beds of starry stonert. Here's some before and after pictures. Obviously the left is a clump of starry, and then after. It's really effective at removing starry stonert, but because we're seeing regrowth, some of the bulbils are still left in the sediment within the same growing season. I just kind of discussed how we managed starry stonert. Now I will be diving into how we monitored starry stonert. We used a variety of tools to try and detect how can we successfully measure success. And basically we did some transect surveys, some point intercept grids, and what we discovered best was just using biomass removal measurements. That was the best tool because some of the point intercept grid surveys, it was just the detection level was just too low to try and find the starry present at these sites. I'm running out of time, so I'm going to just cruise through the results here quick. This is the biomass sampling method we used. We used wet weight measurements. We separated the native plants and we spun the starry stonewort about 20 times before we measured the starry stonewort. Here are results, pretty significant reductions. We did about 2 to 3 hand pulls a year, and our first hand pull in 2017, it does show a huge abundance of starry stonewort, and the last hand pull was just this last August, and it's barely detectable. So really awesome results that we're seeing here. What we've learned, little damage to native plants and macroalgae. It's a really good tool for removing sparse populations of starry stonewort. And basically after 4 years, the starry stonewort has not spread outside the axis and it's been contained. So we were really excited to see these type of results. Not all our projects are this successful. So now I'm going to turn it over to Kylie, and she's going to be talking about Medicine Lake, and that's another lake in Minnesota. Thank you.
All righty. Hang tight for the transition, everyone. And all right, so you should be able to see the PowerPoint, hopefully where Chris left off. Is it looking good, Martha?
Yes, well, it was. Now we went back to what looks like maybe your desktop. There. That's the presentation, or not the presentation view. The
How's
notes
that?
view. There we go. Perfect.
There we go.
And we don't see you, and if that's not possible, that's okay. Just letting you know that your webcam is not on. Oh, there you are. All right, good to go. Thank you.
Yeah. Okay. So my screen's got switched a little bit here, so sorry about that. Let me move everyone over. Alrighty. Also my first virtual presentation, so thanks for bearing with me here. Just wanted to acknowledge Keegan Lund, who is also one of the panelists today. He is actually the senior specialist for my work area in the metro part of Minnesota. So he's on to answer any questions that you may all have afterwards at the end of the session. He was really the lead coordinator and lake manager for this Medicine Lake project. So, all right, moving into Medicine Lake. Is one of the only known water bodies that is in our work area that's infested with starry stonewort to date. It was discovered back in 2018 in late July, and it was discovered by a watercraft inspector staff who found it on a fishing boat after it was coming off of the lake itself. So here are some photos of what the watercraft inspector was able to retrieve. This was then shared with the DNR, kind of normal operating procedures, and then we were able to kind of take some better photos, look at it a little more closely, and verify and confirm that. Here we go. So we worked together with Three Rivers Park District. They actually own the public boat landing as well as the park that surrounds Medicine Lake. So with them, we conducted a lake-wide search, and you can see our boat tracks of the areas we covered in and around this 1,000-acre lake. And as you can see from this map on the left, starry stonewort is mainly in that northern area where the public access is. And here on the right, you have a photo of dense beds of starry. So what that actually looks like when it's covering underwater. So really these specific dense stands that occupy, at least in this instance, about half of the water column. All right, so again, here's a kind of a close-up map of the area that was treated, which is outlined in this green slash area. It's about 15 acres. This lighter green solid color, that are actually bulrush stands as well as water lilies that we wanted to map. Starry stonewort can grow between and underneath those areas, and so we wanted to kind of get an assessment of what would happen in and around those treatment areas. We set up this equally spaced grid. It was about 130 points to monitor both starry stonewort and native occurrences before and after each consecutive treatment. We treated this area about 4 times from early August to the end of September using Chem 1, which is a copper sulfate product, and hydrothol 191, which is an endothol-based product. And so this combination is actually commonly used in Michigan and was what was recommended to us by the applicator. So this map you see in front of you is kind of the spatial locations as well as the abundance from like a 0 to 3 scale, 0 meaning none, 3 meaning a heck of a lot, of starry stonewort prior to any sort of treatment. So this is our kind of initial starting point here. And as I scroll through these maps moving forward, here on the bottom, you'll see a % frequency of occurrence. That's what that FOO is. So basically just % presence of starry stonewort. And then also we wanted to enumerate how many points within our grid we would rate as like an actual 3. And just to give you a little bit of visual of what that is, there's a photo on that left side at the bottom. So yeah, like I said, as I scroll through the next few slides, just kind of pay attention to how the map changes over time, and then that frequency percentage at the bottom, as well as the number of points with 3s. So just flipping back from pre-treatment to the first treatment, we actually saw the greatest reduction in biomass. So those 3s and 2 rate gradings changing from 1s to 0s was during this first treatment. Really saw the drop off there. Here is the second treatment going down to 27% and 7 points. Not changing as much during the third treatment in terms of frequency, but now dropping down to just 2 points with those really high rate gradings. And then finally, this is, you know, the final product at the end of those 4 treatments. So no presence of, you know, really large rate gradings for this species at the end, but still 21% occurrence. So it's definitely still there. It's definitely still alive and present by the end of the growing season. So in 2019, we did this again. We basically saw the same thing. So starry stonewort grew back in the same area. It was just as abundant. And we essentially did the same treatment regime. We did the same monitoring and kind of saw the same thing as results. One additional thing we added to those rate gradings is we decided to add these permanent monitoring stations to measure starry stonewort abundance and those reductions over time. So this is essentially a post that was pounded into the lake bottom. You attach some sort of ruler or measurement, and then we drop down a underwater camera, a GoPro, alongside that ruler and measured the plant bed height within each treatment. So we had 5 stations permanently set up for this. And I'll just show you what that kind of looks like. Apologies for if you get motion sickness a little bit. So dropping down into the deep, and you can see all these little floaty pieces of starry stonewort cells, fragments. And as you hit the bottom, you'll notice that there's actually still plants present in the area that are alive. So one interesting thing is, you know, what you detect on a rake, these double-sided tine rakes, And is that really a good tool for measuring presence, absence of starry stonewort? Because clearly it's there in the video. Oh. All right, the video is going to roll for a little bit longer. I'll keep going. So we also did CET copper concentration monitoring for this project. And basically we saw the same thing every time, that copper really dissipates within 3 to 4 hours of application. So that's just showing that here in this graph. And then one thing we did experiment with was having the applicator, I guess, target certain areas during each treatment. So like for this particular example, you can see he applied more heavily near the bulrush area. And this was basically our way to, I guess, gather some preliminary information on use rates in the field without having those mesocosm studies in place. So through this, we were able to kind of refine or identify some of those higher concentrations that are really needed to produce better results in terms of control. So after completing this now treatment for 2 years pretty intensively, we noticed that starry stonewort creep was occurring beyond the treatment area. You can see that here. We're noticing some points outside kind of on that western tip and then beyond the bulrush beds. So unfortunately, the cat's out of the bag. There's also a couple other occurrences. You see those red dots in and around the lake, quite far off from the actual treatment area where we're now detecting starry stonewort. So moving forward, I think it's safe to say that the DNR and Three Rivers will probably transition into more of a maintenance and control type program where we prioritize those heavy growth areas for starry stonewort. Some takeaways from this project. So with these methods that we used, we were able to effectively reduce the biomass, especially after that initial treatment. Concentrating near the public access, we have the ability to prioritize for those high use areas, and this may potentially reduce the risk of spread to other lakes within the surrounding area. As you know, we've found out that starry stonewort expansion will continue to be problematic, and even though we were able to suppress the starry stonewort in Medicine Lake, we clearly did not completely kill the plant altogether. So that continues to be a head scratcher for us. I think it's important to stress in any sort of evaluation or assessment of your management that the type of data and the type of monitoring you collect is extremely important and can help show that story or tell that story a little bit better. So using things like abundance, rake ratings, as well as any sort of biomass measurement can be really helpful in telling that story. And then finally, I think we just need further research or discussion on, what other tools we have out there in terms of algicides. Should CETs be a little bit higher? Should we incorporate things like bubble curtains to extend that exposure time or increase that concentration? Those questions are to be determined. So just to kind of wrap up some of the work that Chris and I presented, those are just 2 really small case studies of the many infestations and the monitoring that's taking place in Minnesota. I think her and I would both agree that early detection is a really integral part in terms of discovering that new infestation early on, and then with that being able to treat those really small populations in order to contain and just kind of stressing that importance there. So based on what we know so far in Minnesota, pesticides, physical removal, those are both great options, especially for those really small early infestations. And we have seen at least success in being able to hold the line and reduce the spread within the water body. So thank you for that. Like I mentioned before, the 3 of us are all on the panel today and are happy to take questions when that time comes. And if you would like, here's our email contact information. Thanks so much.
Thank you, Kylie and Chris. As a reminder, if you joined late, If you have questions for Chris or Kylie, please enter those into the question box. Sorry, my camera is so glitchy. If you have questions, submit those in the question box, and when we bring everybody back at the end of the session in the panel, we will present those questions to them. Our next presenter is Jen Andreas from Washington State University Extension. Jennifer started working in biological weed control in 1998 at Agriculture and Agri-Food Canada Research Center while completing her undergraduate degree at the University of Lethbridge. She continued with biocontrol research at Kabi, Switzerland, and then completed a Master's of Science in Entomology at the University of Idaho, where she investigated the environmental safety of the hound's tongue root weevil. In 2005, she joined WSU Extension and shifted her focus onto biocontrol implementation and education, with a small component still dedicated to research projects. She is currently the director of the Integrated Weed Control Project. Welcome, Jen, and it looks like you need to bring your presentation up.
Do you need to share, allow me to share?
Quite possibly, let me do that.
It's okay. I still see, yeah, here we go. Okay. And hopefully you can see it.
Yes.
Okay, great. And I'm going to change to the laser pointer, so hopefully that's visible to you as well.
Yes, we see that as well.
Okay, perfect. Thank you so much, and thank you everyone for being here. This is going to be a slightly different topic, talking about biocontrol of invasive aquatic plants. I realize this is a really big topic, and so I'm cramming it all into 20 minutes. So please feel free to contact me if you have any questions about the things I present today. So if you've been working in invasive species management, you obviously are aware of the 4 primary tools that we have in our toolbox for management. And we've already talked today about some physical and mechanical control options, herbicide options. I don't know how often cultural control really gets used in an aquatic system, but I think it can be helpful for replanting wetlands. But what you might not be that familiar with is biocontrol. And Leah had asked me to provide you a presentation on some background information, how we get biocontrol agents into the US, and then what we can expect when we have them available to us. So I think it's always helpful to back up and talk about what is biocontrol and define that. And that's the intentional use of one living organism to control or suppress another. And so you can use things like nematodes or mammals. This is obviously a terrestrial situation. But in classical biocontrol, we typically focus on insects, mites, and pathogens. So there are a number of different kinds of biocontrol, but the kind of biocontrol that I'm talking about today is called classical biocontrol. And this is really based off of a hypothesis referred to as the enemy release hypothesis. So that hypothesis postulates that when our invasive plants come from somewhere else, so most of ours are from Europe, but we do have a lot of Asian invasive plants as well, a lot of those are coming without any natural enemies. And this really gives them a competitive advantage over all of our native plants that have a whole suite of things that are feeding on them and keeping their populations in check. And so really we're looking at these plants having really being successful in their new invaded range because nothing is really attacking them. So with classical biocontrol, what we do is we go back to where the plant is native to it, and we look at what natural enemies are attacking it there. And that might be a large group of things that are doing damage, but then we narrow that down and we try to find something that won't attack native plants or plants of economic value. And so once we do lots and lots of testing, I'll talk a little about that today, to make sure that they're safe, we bring them into the new range, and what we're ultimately looking for is trying to get that balance you find back in the plant's native range. So this is the graphic that sort of shows what our ideally would look like with biocontrol. So I'll run through it with you. On the bottom line, we have time. On the side axis, we have population density. The white undulating line is our invasive plant species. Our blue line is the biocontrol agents. The green dashed line is what we actually want growing in those areas. And the most important line on this graph is really this damaging threshold line, this red line. So before we release a biocontrol agent, we have these invasive plants that are up above this damaging threshold. So their populations may go up and down in response to things like environmental conditions, stuff like that. But the key is we never see them dropping down below this damaging threshold. So at this point, this is a problem. This is causing us problems. So we release a biocontrol agent, and over time, what we want is their populations to build, and then for us to see both the weed and the biocontrol agent populations dropping down below this damaging threshold. So because this is nature and nature never fits nicely into a graph, there's a couple of factors to consider. So the first is here in this pink. So it looks like, okay, we release the biocontrol agents and their populations increase quite quickly, and then we get control. But the time period in here can actually be quite long. So 4 to 5 years, maybe 10 years. So it's really important for us to understand the systems we're working in and to manage our expectations with an understanding that we will get there in the end. The next factor to consider is this period in through here. So yes, it might take 5 years for the populations to build, but it looks like we just get this nice, neat dropping of our weed populations. But the period in through here can actually be really messy and kind of confusing about what's going on. And we really want to think about that in terms of our old predator-prey models we learned in biology classes. So as our weeds are going, our populations are high, the biocontrol populations can get quite high. And then our weed populations tend to do these dramatic crashes. And then of course our biocontrol populations will crash as well. And that gives the weeds an opportunity to resurge, and the biocontrol agents have to build again. And so you can see how that would follow these predator-prey models. The key that we're hoping to see is that ultimately what we'll see is over time, once this sort of craziness comes to an end, is that we get this more even balance down below the damaging threshold. So oh, I'm sorry, I skipped over the third factor. The third factor here you'll notice is that neither the weed or the biocontrol agents ever fall off this graph. And what that's indicating is that we don't see eradication when we have biocontrol as our management strategy. And that makes sense, because if you think about the weed in their native range, it's not completely eradicated by its natural enemies. They're both present. The key is they're below this damaging threshold. So I think we should really expect to see plants present, but not dominating the ecosystem like they were in the past. The other thing I think can be really helpful to consider is looking at what those plants look like in their native range. And so we have this idea that's often tied to the idea of eradication, is we want really to get rid of all of them. But that's not really what we would ever see in its native range, nor is it what we see with our native plants. So if you think about our populations of native plants, we don't have one or two. If we did, we'd have nothing but endangered species. So we have in our native plants and in the native range for our invasive plants, populations that might be quite variable. So in some places like this is flowering rush. We just have a few plants through the system, not very many. But you can also see in Europe fairly large populations of flowering rush. And so we really want to understand that the plants may be there, and they may even be in fairly dense populations, but it's not a system that is not being managed by the biocontrols. So really quickly, biocontrol has a long history. It began in the 1800s with almost no scientific process, very willy-nilly. But that scientific process began about 50 years later, and that process continues to expand and grow and be refined even to this day. Probably one of the most famous and the first biocontrol success story was in Australia on prickly pear cactus. They released Cactoblastis in 1926, and you can see that within 6 years they had some pretty dramatic results. So it can be very successful. In the US, we have our own great examples. This is probably one of our most recent, really awesome examples on Dalmatian toadflax. In 2017, we had a fire wipe through this area. And then within 2 years after releasing this stem mining weevil, we see dramatic reductions in our weed populations. Now, keep in mind, those plants are still there, and I was there this year looking at this site. Plants are still there. You just can't see them like you could initially. So to give you an aquatic example, water hyacinth biocontrol has been really successful in a number of places around the world. There are a couple of different other biocontrol agents on this species, but I'm just showing you today some pictures from Lake Victoria in Uganda. In '96, they did some releases of the Neocotina species, so a couple of weevils, and you can see pretty dramatic results in 2 to 3 years of how those species could wipe out that weed population. We've seen similar results in Florida. In California, it's been less successful, but that's because the biocontrol agents are not very cold hardy, and so they're working towards finding cold hardy strains. So we know it can be effective, but is it cost effective? And what's great about biocontrol is that once those biocontrol agents are established, they really will be self-sustaining. So we have to do very little once they are there and on the site doing work. There aren't a lot of studies looking at cost-benefit analysis, both economically and ecologically, though a recent one came out in 2020, and they're showing economic benefits, benefit cost ratio of 23 to 1 up to 7,400 to 1. And that is the economics. The benefit cost ratio in some other studies that show this includes things like ecological benefits would be 8 to 1 to 3,000 to 1. So biocontrol is very, the cost benefit analysis on biocontrol is really quite amazing. And the final thing that people often say is, okay, it might be effective and it might be cost effective, but is it safe? And are we just releasing the potential for a new biocontrol, a new invasive species? And these are questions we get all the time. What's going to control them? What's going to eat them after the pests are gone? And if you explore the environmental safety record for biocontrol, you'll find that it's actually incredibly robust. A new study came out in 2019 by Hintz et al., where they looked at 457 natural enemies. And of those 457, they found that less than 10% showed some non-target attack. Now, that non-target attack could be something as simple as feeding, so nibbling on plants. But when they looked a little bit deeper, what they found was that less than 1% of those non-target effects actually cause population impacts to that non-target. And so that means we're seeing very, very, very low levels of potential non-target impacts on that would have population level attack rates on our biocontrol agents. They also found that almost all of the ones that were unpredicted was because they weren't tested in pre-release. And as I mentioned, the pre-release studies are just getting better and better. And so we're seeing the incidence of non-targets really decreasing over time. There's a couple of other studies from 2000s that back all of this up. So to get a biocontrol agent into the US, and this is kind of the whole biocontrol process, is a 10-step process. And I'm not going to go through every stage with you today, but I think it's helpful for you to remember that they're not all mutually exclusive from each other. So a lot of our biocontrol programs are in multiple levels of this process at a single time. The first stage would be planning. And so this is where we're looking at what's the feasibility. Is this a good target, potential target weed for biocontrol? Because they're not all are. Not every weed is a good potential target. Are there other people around the globe doing similar work? Who do we want to partner with? And then funding is always the big issue we have. A lot of these programs are sort of stunted by funding availability. Their biocontrol programs are rarely funded by one large source. It's usually involves a consortium with a lot of groups putting in smaller amount of funds in order to have a common goal of a biocontrol agent. So an example of this is parrot feather. In 2020, British Columbia initiated a project. They're trying to find interest for other partners who are interested in trying to find a biocontrol agent for parrot feather. So once we have the money and we're all set to go, then we begin with the exploration. So this is looking in the plant's native range. And they begin that by first looking at the literature and then doing field surveys. So an example of that is Brazilian Eladia. This is a species I'm sure you're fairly well acquainted with. It's pretty bad, and they're doing surveys in South America. They haven't found a lot out there. They did test a fly, but its host range was too broad, and so they're continuing their surveys in Argentina as we speak. Well, maybe not as we speak, but this year. So after you have some potential biocontrol agents and you think you know what you want to work with, then you begin this very critical stage of host specificity testing. So no biocontrol program is going to move past this stage without a lot of work. So this can take years. And really, a biocontrol agent will not be approved for release unless it's gone through very robust testing. And anything that has a too broad a host range is immediately eliminated at this step. So what we're really trying to do is avoid direct non-target effects. So that's the intentional use of an introduced biocontrol agent on a plant species other than the one that we put it out on. And so in order to do this, we have to test a bunch of plants, but that isn't done sort of willy-nilly either. There's a really specific way in which plants are chosen in terms of their likelihood of being attacked by any potential biocontrol agent. And I can go into the detail about that if you're interested. But once this test plant list is developed, then you begin testing the potential biocontrol agents on those plants. And so that's going to involve a lot of different kind of testing arenas and a lot of combinations of kinds of testing. And again, that's an hour-long presentation in and of itself. So an example of this would be for flowering rush. We looked at, there were 6 potential insects and 1 pathogen that looked like they were listed as only feeding on flowering rush. In the end, we scaled that down to only 3 actually look promising. So you can see how you can narrow these things down. Vagus nodulosus right here is a rhizome leaf-feeding insect, and it has been undergoing testing. They've been doing testing where they put a female who's ready to lay eggs onto a test plant. And so her choice is either choose that plant to lay an egg on or not. And if she chooses not to, we can eliminate that insect from the host range. We've got really lucky. This insect appears to be extremely host specific. It hasn't had any interest in any of our test plant species. Another example is with the water primroses. We have 4 non-native water primroses, and unfortunately we also have a lot of native water primroses, which might make things quite difficult in terms of finding a biocontrol agent. So they've been conducting surveys in South America. They've tested 4, but they had too broad a host range, and so they're now moving on to testing this stem mining weevil for the primroses. So it's okay if we see some feeding and egg laying. That's fine. The plants, that's not necessarily plant usage. It gets a little more concerning when we see some larval development, and we're particularly interested in anything that can complete development on those plants. That means going from egg to adult on those plants. All of the species that fall into this category are studied very intensively. So I won't say too much about this, but I think it's really valuable for everybody to understand that all biocontrol agents have to go through an approval process through the federal government, and that no biocontrol agent is ever released, not even for trials, without having gone through this process. And I'm not going to run through this with you, but I just really put it in here so you can see that these decisions are not made lightly. It usually takes a minimum of 3 years before anything is approved for release. So then we get down to, once we have it approved for release and APHIS has said, yes, we feel this is a safe agent and should be put out in order to control our species, we begin this these are sort of 3 that come together: rearing, colonization, and establishment. And a really great example of this is our new knotweed biocontrol agent. This is a sap-sucking psyllid, and there are 2 different strains attacking the different knotweed species. We released these in 2020, so pretty exciting project. And this is some of the damage that I found on our giant knotweed by the psyllid. And this is a picture of the psyllid, so you can see how tiny they are. So we're just in the very beginning stages of implementation and understanding what this will do in its introduced range. And finally, we have evaluation of biocontrol. And this is really probably the most critical thing we can do, because what it tells us is whether we were successful. Unfortunately, there's rarely any funding for this kind of thing because we move back up to here with another species. But evaluation is really critical. So we can say, was this biocontrol agent ultimately effective? I don't have a lot of great aquatic examples of that of this process, but I did want to mention purple loosestrife. I'm sure you're very familiar with this plant, and we have a couple of different biocontrol agents. But the big player in all of this is the two Galeropsella species. They were first released in 1992, and we saw widespread success across the US. So this is a before and after pictures that I took up on the San Juan Islands. It's a small pond, and in the left pictures are the before. And it's hard to see, I apologize, we took pictures in the spring, but trust me when I tell you that all of these plants in through here and all of these little plants in through here, that's all purple loosestrife. We come back a couple of years later, everything that you see that is brown is fed on purple loosestrife. The beetles completely skeletonized it. So this kind of shows, it's a nice graphic to be able to demonstrate the effects of biocontrol in the long run. So really, biocontrol can provide a long-term sustainable management option for land managers. Unfortunately, it's often considered as sort of a last resort. Usually the weeds are out of control and people say, you know what, we should start a biocontrol program. But biocontrol programs take a minimum of probably 10 years. And so that's unfortunate because then the weeds have an opportunity to really take off and while we're searching for a biocontrol program. Biocontrol is really an important part of the integrated weed management toolbox, and I think they're all equally valuable tools. And it also has a place in aquatic systems. So this is my contact information. If you have any questions, I would love to hear them. And if you're interested in more biocontrol information, we're going to have a biocontrol summit at the end of October through NIESMA. So you can contact me about that as well. Thank you.
Excellent. Thank you, Jen, so much. And again, if you have questions for Jen for today, please enter those questions in the question box in the webinar, and we will get to those at the end. Next up, we have another co-hosted presentation by our friend Dennis Zabaglo and John Paluccio, if I get that right. So we'll begin with an introduction of Dennis. Dennis is the Aquatic Resource Program Manager and a Principal Environmental Specialist at the Tahoe Regional Planning Agency. Dennis has over 25 years of experience in natural resource management and leads Lake Tahoe's robust aquatic invasive species program that includes dozens of partners, both public and private. His commitment to fostering partnership education coupled with strategic thinking has allowed the program to become a national model. In his free time, Dennis enjoys all of Lake Tahoe's splendor and cheering for his beloved alma mater, Penn State. Welcome, Dennis.
Thanks, Martha. Unfortunately, it's a little bit smoky here today, so not really able to enjoy those outdoors that we take advantage of, but wanted to kind of just provide a little bit of background information. John Paluccio, he is the principal for Inventive Resources Incorporated. Sorry, John. And he's an inventor and has been working with us for several years and has developed this UV light technology to treat invasive aquatic plant species. We've been doing this for a few years now. Some of you may remember a presentation John and I gave at the San Diego meeting that was, I guess, the friends of the Western Regional Panel, since we were on a strategic pause then, but we've been moving forward with testing this technique. Tahoe has designation of an outstanding national resource water, one of only two in California, the other being Mono Lake. And with that comes very strict anti-degradation policy requirements that even apply to restoration efforts. And so when we are treating invasive aquatic plant species, aquatic herbicides have never been used in Lake Tahoe and until recently have been expressly prohibited. And so we try to look at innovative approaches to build the toolbox, if you will. And so John has been adapting this technique, and we'll be showing some updated results and the progress we're making there. But in the middle of the presentation, I'll kind of jump in again and show how, or talk about how we are including it into a pretty large scale test of this, along with aquatic herbicides that's been proposed by a private entity that owns quite a bit of waterways connected to Lake Tahoe. And so I'll turn it over to John Paluccio, and he can provide the update on the progress we're making with UV light technology. Thanks. Go ahead, John.
Okay, great. Let's see, get my screen shown here. Is my screen showing?
Yeah, we can see it. We might just need you to put it to, yep, there you go.
Okay, great. And am I above the picture also, or is that? Okay.
Yes, you are. You
Okay.
can see your webcam and your presentation.
Excellent. All right, so this is going to be an update on using UV light as a tool and to treat aquatic invasive plants here in Lake Tahoe. So the project partners are TRPA, the University of Nevada Global Water Center, Cardno, Inventive Resources, Lakeside Marina and Beach, and the Tahoe Keys Property Owners Association. So we're going to talk about the objectives of the project, the equipment used, locations, and so the first slide here is where we did our treatment. So we tested the technology in this marina, which is Lakeside Marina, and then below is a swim beach. And this beach is used by swimmers every day. It's a very popular beach, but you can even see in this image, there's an area of dark plants right in the middle here. It's been our focus. On the right side is Tahoe Keys, and we are focusing on this lower section, which shows a blow up. It has a combination of a lot of boat slips, very deep areas and shallow areas. The project objectives is to expand our knowledge using UV light to treat the aquatic invasive plants. This is a continuation of the project we did in 2016, 17, 18 with TRCD, and that publication can be found online on their website. But our goal now is to treat even larger areas. From back then when we did like a half an acre, we're now doing up to almost 3 acres of treatment, and being able to test the plants at different growing phases. So originally we really came in July and treat them when they're more mature. Now we've got a chance to treat them from the very early stages of development all the way through to fall. And so we're also going to determine when is the best time of year to treat these plants. So equipment used. Back in 17, when we started this project, we had a very small array. We came out there with a 4-foot by 8-foot array. We learned very quickly we'll never get this project done with something that small, so we immediately expanded it to something around 5-foot by 16-foot, and that allowed us to finish that project. And that system ran off 4 propane generators. Then in 2019, last year, we built a new boat, and it had an array that was 8-foot by 20, and it was powered by 6 propane generators. And after doing that trial, we realized, okay, still, we need to bump this up a little bit more. So this last winter, we built another boat, and it has an array that has an array of 12-foot by 32-foot, and it's powered by a 99 kW diesel generator. And we're really only using half of the power off that generator. I can make the array almost twice as big, which would really increase decrease treatment time and speed up the process. But that's something we hope to do in the future. Phase 4, which is next summer, we learned a really good lesson about having an array that was 12 by 32, and that was that you really couldn't clean it that easy. It forced us to really work with working underwater, which is not the best thing to do. Is to try to clean the lights underwater was difficult. So we're going to go back to something more narrow and streamlined, but make it much longer so we can treat and stab underneath some of those boat docks and treat under the slips. So that's our goal for next year. And in the future, if we are put to work out here in Tahoe and other areas, it'd be great to build something larger like the image shown here, where it's a houseboat, where you could have a 20 by 40 foot array, and that size unit can treat an acre in 1 to 2 days. So that's really going to be the goal in the future as we move forward. This is a schematic drawing of what was built this last winter, and we decided to cantilever the array on one side over 4 feet, and that was to enable us to treat underneath the slips easier. The generator is placed right in the center of mass of the boat, and then the fuel on the backside. We did add a cabin for the crew. After working all season last year, we realized weather is tough, so we wanted to make it a little more comfortable for them and allow them to take better notes and you know, more comfortable on the boat. This is some photographs of the final unit in action. That top image is taken at Lakeside Marina off one of the taxi walls. It's a very busy area. Always has plants growing there in the left and the right hand side of the image. We did have a second deck, and that allows us to really look down and see the plants and see any obstructions. The pictures on the bottom are of the boat working in the beach, and you can see some ropes in the background, and I'm going to go into that, how that was very helpful with treating the entire beach in an arc movement. So one of the first things we do when we tackle an area is we really survey it. We take a lot of aerial photographs. We take measurements of the sediment height, and we sort of came up with a treatment goal. And this was our treatment plan for the beach and the marina. We then calculated the area. It was a little over 2 acres of treatment, and we set to work. This is going back to how the ropes were attached to the boat. We tied off the 3 points on that wall, and this can also be done with just a stake in the ground. We've experimented just, you know, taking an auger and putting it in the ground and working the same concept in an open water condition, but it was nice having that wall for this project. And we just basically treated in this arc form, and you could lift the array up, move over, say 30 feet, drop it down, just so you're above the sediment level layer, and treat the plants. And we did the whole beach this way, and in the marina. Here's a few photographs from the project last year. This is 2019, around this time. And you can see the majority of the beach was covered with chara, but you can see a few dark areas in the center. Not the shadows, but the plants. If you look carefully, you'll see them a little poking out of the chara. And those are our really important areas we wanted to target. This is a couple months after the first treatment. We came back to assess the damage that we did the first time, and we still see some dark areas where we didn't hit them again in late October. This picture is from January. Pretty much the beach looks pretty clean. You can still see in this bottom corner here, you can still see our project site from 2017. It's still visible where we treated the first time, but for the most part, beach is very, very clear. A couple months later, we're still monitoring, and in June, we start to see there's a few areas that are starting to look like there's more plant growth, which those are our target areas for this year. And we did put these markers down. We've actually 4 down for the divers to really analyze those locations and compare treated and non-treated. And it just helps monitor the beach better this way. Now going back, I want to take a picture back from 2017. This is what the beach used to look like. The plants were much taller, much more curly leaf throughout the beach. And those plants are, you know, this is a picture. This is a 4, 5, 6 foot tall curly plants loaded with turions. This is where we first did our project in 2017. Just wanted to show what we were dealing with then. Then we had 2 years of UV treatment. And this picture was taken back in August before treatment. Same exact location. We did not identify any curly leaf. There was milfoil and chara, and that's throughout the whole beach. So that was a real benefit this year, and that's what the UNR has been studying the plants growing in this beach, and they've identified what types of plants and how many of each. So I added this slide. I just wanted to show you the performance of how UV affects the plants. This is before treatment. Typically takes about 2 weeks after UV treatment for the plants to drop, and you can see what they look like after treatment. 2 to 4 weeks later, the plants turn into this sort of spongy material. It's a very slow process for decomposition. It's not instant. And then after 6 weeks, you could take your paddle and it basically just turned to a fine dust. And so I was like sharing this slide to show the process of treatment with UV light. And that's where University of Nevada is coming in. They're doing the science behind this project. They're measuring plant composition, the plant biomass coverage, height of plant, and water conditions. And they're also doing a lot of, the last 2 years, they've been working in the lab, studying the plants. In a lab condition, they have about 300 vessels here where they're testing UV light on different species at different times. And right now they're focused on the turions. How does UV light affect the turions? And we're learning, the pros and cons of that. Really, UV light really doesn't have a penetrating power, so it really doesn't want to penetrate anything that is kind of surface. But translucent plants, it works much better on the thin, delicate aquatic plants. So this is a method we came up with just for quick analysis of how you can treat the plants and monitor and see how the system works. We drop these sandbags right on top of the aquatic plants, and then that way we could identify them on the GPS of where we put them. We then drop the array on top of the plants, and the marker's right around this area. And this is what it looks like 2 weeks after treatment. Pretty much decimated the plants in that area. Everything turned to pretty much a dust color. The few plants remaining, we would then hit on the second treatment about a month or two later. But I think this slide really shows the effect of UV light on these smaller, more delicate plants. And really the goal here is to really disrupt the growth cycle. So if you did have a plant that was 2 or 3 feet tall, ready to mature, ready to drop turions, and you hit it at this stage and you don't let it produce the turions, you really can stop that growth cycle. And that's something that's really important to realize is using UV light, hitting the plants young and disrupting that growth pattern, they're not going to come back that same year. The water conditions are different. It's warmer temperatures. They don't grow as fast. So there's some benefits to treating early and stopping that growth pattern. This is a image of using barrier mats in conjunction with UV light. So the mat company put the mats down right at the entrance to the marina, and sure enough, plants grew right on the other side of it. We then treated these plants right after this image, and this is a couple months later, and you can see when the mats were removed, the difference between the two sites look very similar. And now the same exact location is being monitored a year later to see the grow back when comparing both mats and UV. This image is a lakeside marina. We worked with the local agencies, and they were able to put mats around the perimeter, and that was very helpful for us. It allowed us not to have to get so close to the edges and allowed us to cantilever the array underneath the boats and move forward. And so that saved us a lot of time using a combination of technologies to treat using UV light and the bottom barriers, and allowed us to get a very thorough treatment in this marina. And again, this image is how we basically rotate the array 90 degrees. We have floats on it and weights, and allows us to stick underneath these areas and drop it down 8 feet just right above the sediment layer and just move forward real slowly. This last season, we really learned when's the best time of day to treat, and we've learned that 5 in the morning till around 11 a.m. was great. After that, there was so much boat traffic, a lot more wind in Tahoe, that we decided to we would like to break into 2 shifts, have a morning crew and an evening crew. And if I ever had, a larger vessel, we would be able to do that. But this is sort of the goal, is to treat in the evening. It's very calm. There's not a lot of boat traffic, and the water's a lot clearer. So it allows you to see underneath the array. So this is a short little video clip of the work done in the marina. And this first image is just 20 days after our first treatment this year. So we dropped the camera down about 7 feet and you can see the plants are already decomposing. Sort of looks like the beach image. These plants were not treated yet, and they're 4 to 5 feet tall curly leaf. If we didn't treat the marina, it would all look like that. And we turn the camera back around 180 degrees, you can see underneath the boat slips. And take one more shot here of some nice looking fish swimming by the camera. But just to give you an idea of what a treated versus a non-treated area looks like, we then came back 3 months later and just took this last week. And you can see we had a little bit of grow back. There's a few plants growing here, but for the most part, very controllable for the season. And that's after the first treatment. We're now going to go back in October and treat a second time and pretty much knock out all the plants in that marina. And this is where I'll introduce Dennis back to talk more about the Keys project.
Yeah, thanks, John. And so I mentioned the Tahoe Keys, and I think many of you might be a little bit familiar with this location if you've come to Tahoe for any of the previous WRP meetings or the Aquatic Nuisance Species Task Force meeting, where we've gone and taken a look in here. But it's on the South Shore, pretty much right at 6 o'clock. If you're looking at Lake Tahoe as a clock, it was once part of a wetland, as you can see on either side. The marsh systems there, it was the largest wetland in the entire Sierra that was turned into this private property residential development back in the 50s. And so those waterways there's 2 channels connecting directly to Lake Tahoe. These are considered to be waters of Lake Tahoe and waters of the state and waters of the US. And so carry that anti-degradation policy I mentioned previously. But it's about 170 acres total of waterways, almost entirely infested with Eurasian water milfoil and curly pondweed. And some of the larger control projects we've done to date in the lake are about 6 acres. So now we're looking about 30 times larger. And so some of the more mechanical methods we've used in the past with bottom barriers and diver-assisted suction and hand pulling just really can't keep up at this scale. And so looking at new tools like UV light, but the Homeowners Association here has proposed the use of aquatic herbicides for the first time in any water that's considered an outstanding national resource water. So we're going through a very robust and complicated environmental analysis along with the anti-degradation analysis that's required by the state if herbicides were to be used. But looking at a test method that looks at multiple treatment methods to determine what might be the best collection of tools for looking for dealing with the infestation as a whole. So right now we have about 20 some sites at about 41 acres of the entire 170. And if the polygons that you see on the map are just different types of controls. So looking at herbicides alone, looking at UV light alone, and looking at both of them in combination. And if John, if you can go to the next slide, the combination approach is trying to maximize the effectiveness of both tools, both UV light and herbicides, while minimizing any potential use of herbicides. So John will show you a picture here in a moment or two, the difficulty it is leading up to edges, especially in a situation like this where we have channels that have different bottoms, they have sloping sides that the UV light array may not be able to treat effectively, at least at this time. And so looking at using UV light up the middle of these channels with herbicides used on the edges with potentially turbidity curtains, or as I think you heard Dr. Sartain talk about earlier, potentially bubble curtains. I think there's a test that's going on in the Northwest that's looking at bubble curtain used to contain or to hold herbicide concentrations. But this is kind of what we're looking at. The environmental document just was finished its public comment period for the draft environmental document, draft EIR, and looking to have those certified early next year with potential treatment with UV light and potentially others like herbicides happening in the spring of 2021, early in the season. Has been pointed out that treating plants early when biomass is less is more effective, and from a nutrient release standpoint, also better for water quality. So I'm going to turn it back over to John to talk about what he's doing here in the Keys to investigate his tools to ensure that his equipment is going to be the most effective in this situation.
Okay. So, am I back on? Everything good? Okay. This is the keys map, and we created this proposed treatment map, and University of Nevada chose these transect lines to study. So the red is where we wanted to treat with UV. The yellow are our controls. And we also added a lot of sections up here at the top that had a lot of obstacles, boat slips, working under the boats, working under tight docks. And so we added these just to learn how we're going to work in these different conditions. This picture was just taken last week. This is about 60 days after treatment. These are parallel transects where we treated back in June, end of June. And this picture was just taken last week. So it definitely had a good effect on the young stages of the plants. And this next video clip is exactly that same location. I think it's just good to see an underwater view of what the plants look like after treatment. So that's that first side-by-side transect. Again, we just dropped the camera down. Sorry, the water's not super clear here, but you can see as it goes down, it sees the dust cloud, and there's really no evidence of plants where we treated. We're then going to lift the camera up, go to the other side, and it's just loaded with the milfoil, coontail, and curly pondweed. So it just gives you an idea of about 60 days after treatment. And we just brought the boat back about a week ago to do another round of UV treatment at the Keys. So we're talking a little bit about when's the best time to treat. Really, the advantage of using UV light, you really could do it year round in some areas, but in Tahoe, due to the ice and the super cold temperatures, we decided, anytime between May and October are the best times to treat using UV light. We do know if we can treat early, in these early stages, we get the plants when they're very tender and young. As you get up into September, August, September, October, they're getting more hardy, takes a little more treatment time. We sort of missed that opportunity, but we can still knock them down, which is good. And then always doing a late fall treatment, get some of the early risers, like curly leaf sometimes sprouts very, very early in the season, and then waits until spring to really explode and grow. So we have seen evidence of curly sprouting this late. And if you can hit it when that's that young, that can be a real advantage. Sort of like this picture, we were able to bring the boat as close as possible to the wall. And this was again taken in June. You could see how dense the plants were. And this was taken 2 months later. And pretty much everywhere we treated shows very, very little plant growth. But where we didn't, this was taken in August, end of August, it's very dense and thick. So I am also open for questions through the panel, and I'd be glad to answer any at the end of this presentation.
Great, thank you very much. We're going to go ahead and move into the Q&A now. So if all of the presenters could pull up their webcams, and we will get the panel discussion started. Okay, so we'll start off with Dr. Sartain, who presented first. The first question is from Linda Shaw. Have bubble curtains been used in tidality influenced wetlands or estuaries? Oh, you're muted there.
Hear me now?
Yep, we can hear you.
They have not been used in tidal areas for increasing herbicide exposure in high water exchange areas. This year, there was some talk of possibly doing a demonstration in the California Delta, where, you know, they have some pretty significant tide swings there. But as far as I know, as far as increasing herbicide contact time, they have not been used, or there's no research out there showing their impact to water exchange when you're in a tidal system.
Okay, thank you. So I will just give all of the questions for the starry stonewort folks. I'll just present it to all of you. So was there any fish mortality observed associated with the copper and hydrothol treatment? From Tom Wolf.
We didn't do any sort of fish assessment at all for the copper treatment. However, we didn't see any sort of even small fish floating. There's actually a turtle breeding ground with, you know, adjacent to the treatment area, relatively close. We monitored for that and we didn't see any mortality of that as well.
Okay, great. Any other additions to that? All right, the next question is for Jen. For the less than 1% non-target impacts, has the severity of the 1% been investigated? From Tom Boos.
Oh, great question. I am not positive on that. I imagine there are ongoing studies, but a lot of those, a lot of this research is from, you know, decades. And so the details of that, I don't know the details. If you would like to send me an email, I could look it up for you.
Great, thank you. Okay, for John and Dennis, are you gathering any non-target species effects or impacts of UV on fish, reptiles, and amphibians, sediment, etc., in the Northwest? Endangered species such as the salmon are of great concern as to presence during treatments and effects the treatment method will or could have on adults and juveniles and their food chain. From Damian Walter.
Yeah, so we are doing pre, during, and post-project monitoring for different species. The fish themselves are seen to scatter away, and we have not seen any kind of impacts to any fish species. But with non-target plant species, they are certainly affected, as they would be with other methods that we've used with bottom barriers. What we've experienced in Lake Tahoe, at least, is that while any natives that may have been impacted generally come back stronger, and with benthic macroinvertebrates, we do see a temporary decline in their populations, but then rebound very quickly to sometimes higher numbers than pre-project after a certain period of time post-project. We have some initial monitoring from the pilot that was done in 2017, and I can share that information that has some more specifics on that. And as I said, we're doing that with this pilot as well.
Anything to add to that, John?
Something that some people don't realize is actually a box. We actually drop a box over the plants, and so the 5 sides are shaded. The light's only directed straight down at the plants, and so that's something that we've got many photographs where the fish are floating, just swimming right above the array, and you know, not affected at all because we have polycarbonate and UV light does not penetrate anything, you know, through the water. It's only about 6 inches to 12 inches of penetrating power. So the plants are only treated underneath the array.
Okay, thank you. The next one is for Dr. Sartain. Is there a depth limit beyond which the bubble curtain will be ineffective? From Aaron Murphy.
We have not put one out any deeper than about 15 feet. So as far as I know, the deeper you are, you're likely going to have more air power or a larger compressor. Just as if you're going to have a longer curtain, you would need more a larger compressor to make sure you have even distribution throughout the curtain. But as far as I know right now, based off what we've done, we haven't seen a depth restriction based off the bubble curtain. I'm not sure if maybe if you're talking about 50 feet deep, there has been some talk about using it offshore with some dredge operations or some sound abatement for drilling off coasts for oceanic mammals and stuff like that. I'm not really too aware of what they've seen there, if there's been a depth restriction, but based off our research, once you get deeper than 15 foot deep, you know, that's kind of out of the littoral zone or that 18 to 20 foot deep area. So we're trying to maintain it in just the littoral area. So I guess unless we're at Lake Tahoe where the water's gin clear like that, anything over that 15 to 18 foot mark really wouldn't require a bubble curtain.
Okay, thank you. For Chris and Kylie, were folks okay with having a permanent marker in the lake? Boats were good about not hitting them, or were folks removing them?
So for our permanent posted stations, we put those orange mooring buoys on top of them to mark them for navigation. We didn't have any issues with folks moving them, and when we could, we tried to create established stations on permanent structures. Like one was attached to a sailboat dock, and so we really tried to be considerate of any sort of navigation that would impede. We're also talking about this treatment area. The average depth is about 2.5 feet. So typically boats aren't going to this area to begin with. And it's on a kind of undeveloped shoreline too. So we didn't have any issues there. Thanks.
Great. Oh, Chris, you're muted. There you go.
Thank you. I was muted by the organization, so I didn't have anything to add. We didn't actually put any post in the lake. It was just the day of, we put out the buoys.
Okay, great. Thank you. The next question's for you, Jen. Could you comment on the use of standardized impact monitoring protocol? How generally accepted and utilized is it, and how it can best be used?
Sure. So the SIMP, or Standard Impact Monitoring Protocol, is a way for us to look at long-term changes in the system after a biocontrol agent has been released. So it's a really simple, it's designed to be really simple and fast, because monitoring protocols can get really time consuming, and then people are less inclined to do them. So this is designed to be fast, and the idea is that if we have a lot of sites, there will have statistical strength by number of sites. And so that's a protocol that's being used by a lot of people across many Western states, but only for terrestrial environments. So that's sort of been narrowed down for terrestrial environments. So it hasn't, as far as I know, been used in aquatic systems. And so I think monitoring for biocontrols is going to be really challenging in an aquatic system. And I'm taking notes here because I think there's a lot of opportunity to learn things about how we can monitor when we start releasing, say, the flowering rush biocontrol agent. So I'm taking notes on different ideas you guys are using and just trying to sort of brainstorm that. So I think the SIMP, there will be components we can put into an aquatic system, but I think we'll have to modify it, because obviously that's a much more difficult and different environment.
Okay, thank you. For John and Dennis, two questions here. Are some species more susceptible to treatment than others? And then the second question, could the UV treatment boat be scaled down to a smaller unit to spot treat smaller areas with infestations?
So the thinner the plant, the more fragile the plant, the faster it does drop. So the leafy pondweed was one of the first plants that usually drops in the system. I do have another graph, and a lot of this is on the other study that we explained in our findings. But the thicker the plant, like the Richardson, it has a thicker stalk. It does take a longer treatment time. They all drop over time, but you may have to increase the treatment time slightly from 5, 10, 15. It really depends on how many minutes is needed or how many treatments are needed. So to answer the first question, yes, the plants do drop at different rates. The more fragile, like coontail, leafy pondweed, the milfoil are all in the same range. Where you get into the curly leaf, Richardson, a few of the other thicker plants, they do take a little more treatment time because you're sort of penetrating that cell structure, and it takes longer for the plants to drop. And then what was the other question?
Could the UV treatment be scaled down to a smaller unit to spot treat smaller areas with infestations?
So I also took that slide out, but yes, we did make handheld units. And the problem is, I guess if it's a very small area, yes, you can hand treat it. But being in these conditions, we just found it better to scale up, so we're not there as long. But yes, it's just a UV light on a pole. We've allowed it. We went down to go put it in areas where the wall had these depressions, and we tested that on the beach side. But you just got to sit there for 5 minutes. It's like, why not build something 4 foot by 4 foot or something larger to treat a larger area? But yes, it's scalable, absolutely. We just rather go bigger.
And just to tie off of that, John has also been investigating use of like a mat, flexible mat, because of that area I mentioned within the Tahoe Keys. It has kind of a convex or concave, it's a channel. So there's curved bottoms, and so some have riprap, some just have slope dirt walls. And so if there's an opportunity to advance the technology to deal with those kind of areas where the boat itself has a difficult time of reaching, that's something he's also looking at developing.
Great, thank you. The next question is for Dr. Sartain. Is the bubble curtain fairly low impact on the lake bottom, specifically regarding potential disturbance to cultural resources?
So once you initially put out the bubble curtain, you might see a little bit of sediment come up when you initially power it up, but other than that, it's no real impacts. If you have a pretty small team to deploy it, and there's very dense plant material in the site, you will catch some of those plants on the tubing itself and pull up some plants that way. But as far as increasing turbidity and having an impact on the lake bottom, the tubing itself is very small, 3/4 of an inch or 1 inch. So it would compare, if you went to the hardware store and saw the size of a 1 inch PVC pipe or 3/4 inch PVC pipe, that's about the diameter of the tubing, and the bubbles actually come out at the top. So you're not getting any airflow directly into the sediment to cause turbidity issues or anything like that.
Okay, thank you. And actually just one more question for you, Dr. Sartain. What would you expect the results to show if a traditional curtain were used for containment versus a bubble curtain? For example, in areas where the benefits of using a bubble curtain aren't recognized, do you expect that a traditional curtain would be more effective?
Yes, actually, I didn't have enough time to present some of that data today, but that site where we did the bubble curtain research in 2018 and 2019, we put a traditional barrier curtain at that site to see the differences between the two. So if you remember from my first 2 evaluations, the half-life of the dye with the bubble curtain was about 7.5 hours, 7 hours. With the barrier curtain, we were looking at about 32 hours. So if you can imagine, that curtain goes all the way to the lake bottom, and there's very little areas for that water to move back and forth between that treatment area, except some very small seams in between where the curtains are connected and along the shoreline. So we expected the traditional curtain to work fairly well, and it did outperform the bubble curtain, but it's kind of like a cost benefit deal where with the bubble curtain, you're not restricting traffic navigation of that matter. It's actually pretty neat to watch. You can see small ducks and stuff pass through it. We've even seen fish hang out around it, where with a barrier curtain, the amount of labor it takes into deploying one of those, particularly too in a river the size of the Columbia, when you have that much current going out there, and you have 15 foot of curtain going down to the bottom when you're putting out that curtain. If you get that current wrong, it can kind of put you in a tight spot. But
Okay, great, thank you. And we did have a few more questions for the UV folks, but we'll go ahead and circulate those through an email and get a written response for them, because we are running up right to the end of time. And I'm gonna pass it over to Martha to close us out. So thank all of you who are attending, thanks all of the presenters, and thank you for taking some time to answer these questions.
Thank you.
thank you, enjoyed it.
Thanks, Mason. Mason, do you have a couple of announcements regarding the other extracurricular activities after this?
Sure. Yeah, so at 5 o'clock this evening, we are going to be having a happy hour. That's 5 o'clock Mountain Time, and there's a Zoom link that's been circulated to everyone who's registered, as well as posted up onto the WRP 2020 virtual meeting Facebook page. There's a link to that on the WRP's website, and you can also find it by searching WRP 2020 virtual meeting on Facebook. If you're having any trouble accessing that, just shoot me an email and I will get you a link.
Thanks, Mason. All right, well, thank you everyone for joining us today. We hope you'll join us the rest of the week. Tomorrow is invasive fishes and eDNA, and on Friday is our business meeting. So everyone is welcome to attend the business meeting as well. So with that, have a great evening, afternoon, and we hope to see you tomorrow. Thank you.